नाउ देयर इज अ एनिमेशन फॉर यू। ये जो एंजाइम यहाँ पर प्रेजेंट है, it is going to interact with this substrate. Suppose this is a substrate. This substrate will be came here and will be fit in this place. After that ES complex will form. So this is the substrate. The portion or the moiety which is going to interact with the active site of an enzyme must be complementary with the enzyme's active site. ये कार्टून में देखो, the active site of this enzyme is looking like that. This substrate have the complementary binding site is like this structure. So it is never possible this substrate can came here and interact. यहाँ पर आएगा लेकिन interaction possible नहीं है. So it will be unable to interact with the substrate. This is very important that the structure which is going to interact with the enzyme is equally important like active site of enzyme. Now there are two hypotheses about that how a enzyme substrate complex can be formed. So first one is lock and key hypothesis. देखो क्या होता है यहाँ पे. Suppose this is a enzyme and this is a substrate 1 and there is another substrate. This is substrate 2. This substrate 2 is not able to interact with the enzyme active site. Because why? Because this portion of structure, this portion of this substrate is not complementary with the enzyme's active site for interaction. But this substrate 1 can easily came here and interact with the enzyme. देखो कैसे होगा, ये आएगा और यहाँ पर इंटरेक्शन हो जाएगा. So by this method the enzyme substrate complex is formed. So from this hypothesis it can be concluded that the active site is rigid structure. Why? Because if the active site is flexible, if it is flexible, then substrate 2 can also be accommodated in the active site of enzyme. But if a structure is rigid, then only the structure which have compatibility with this can able to interact. So active site is rigid according to lock and key hypothesis. Now we are coming to induced fit model. This is very important and this is the main concept upon which total enzyme is dependent. So suppose this is an enzyme. There may be a substrate 1, another substrate 2. Now according to you, which substrate is able to interact with the enzyme? You can say that the substrate 2 is able to interact with the enzyme, but it is not. It can't able. Why? I am coming in this portion. There are some charge. Which charge? The charge of R group, which I say previously. The charge of this side chain of amino acid. There is C double O minus charge. There may be a plus charge of NH2 group. So these charge play crucial role in case of enzyme substrate reaction. So suppose there is plus for which amino acid? It may be for lysine, or it may be for arginine, or maybe for histidine. Suppose there are 2 plus and 2 minus. and this substrate 1 have also minus, minus, plus, plus, and this substrate 2 have plus, plus, minus, plus. Now this is a electrostatic reaction. The plus charge will be interact with the minus charge, and the minus charge by same way will be interact with the plus charge. Between same charge there will be present a force known as repulsion. So based upon this concept, you can easily say that this substrate 2 is never be able to interact with this enzyme, but this substrate 1 can easily came here and interact with enzyme and can form the enzyme substrate complex. So from this induced fit model we can conclude that, देखो यहाँ पे एंजाइम का जो एक्टिव साइट है, this active site is totally different from the site which is going to interact with S1. So it is never be possible the interaction between E and S1 without the electrostatic interaction. I mean, I want to say that the active site is flexible. That's why this interaction is only possible. So active site is flexible. Electrostatic interaction, H bonding, I mean all non-covalent interaction are the main or major force for these enzyme substrate complex formation, and this is supported by induced fit model. Hydrophobic interaction and van der Waals force. This is the induced fit model. I think you are clear about these two model, which is lock and key hypothesis and induced fit model. Now I am coming to part by which enzyme is formed. So enzyme which is a protein is formed by two parts. One is apoenzyme and another one is cofactor. This apoenzyme is the proteinaceous part for which we can say that enzyme is a protein, and this cofactor are of two type. It may be a metal ion or ion, it may be a ion, or it may be a coenzyme. The coenzyme which is bound with the enzyme complex tightly. I mean, the example of coenzyme is, okay, I am coming in that part later. Okay, this may be coenzyme or prosthetic group. Now the example of ion is, it may be zinc 2 plus, it may be Cu2 plus, it may be Ni2 plus. Zinc 2 plus act as a cofactor in case of carbonic anhydrase. This carbonic anhydrase is one of the fastest enzyme. This will be the proper word, because catalyze is also the fastest. Mainly, I mean, catalyze is the most fast enzyme. So carbonic anhydrase is needed a cofactor as ion or metal ion is known as zinc 2 plus, and Cu2 plus is needed by cytochrome oxidase, which is an enzyme of ETS, electron transport chain. And this nickel 2 plus, this is an enzyme of, this act as a cofactor of urease. ठीक है? And if we came about the coenzyme. These coenzymes are mainly vitamins. I mean mainly the water soluble vitamins. Water soluble A, D, E, A, D and E, vitamin A, vitamin D and vitamin E will not behave as a coenzyme because they are lipid soluble or fat soluble. So here you can see that this is the apoenzyme which is remain bound in the coenzyme. When apoenzyme is bound with the prosthetic group, there is a strong interaction, so that here this strong interaction is denoted by 4 lines. They never leave the enzyme complex for interaction with other substances. When we learn about the TCA cycle, then it will be clear for us that how the prosthetic group is actually worked. Okay. Now, and in case of apoenzyme and coenzyme, you can see that there is only one rod by which they are saying that this interaction is not as much as stable like prosthetic group. So what is the example of coenzyme? The example of coenzyme is vitamin B1, which is a TPP, which act as a coenzyme of pyruvate dehydrogenase. Okay. Next is vitamin B2, which is riboflavin, and like that. So here is a chart is provided for you, which I already discussed. This is a Cu2 plus, which is for cytochrome oxidase, and K plus, and for Mg2 plus, and for MO, and for Ni2 plus is urease, and for Zn2 plus it is carbonic anhydrase, which I already told you. And there is a chart is provided for you. You have to just learn the name of coenzyme and the vitamin which is from which it is actually produced. Okay, so there is coenzyme A, and there is nicotinamide adenine dinucleotide NAD, which is produced from niacin, I mean vitamin B3. It is vitamin B3, and flavin adenine dinucleotide or FAD or FADS2 or FMN, like that, they are produced from riboflavin or vitamin B2. Pyridoxal phosphate PLP, which is needed for amino group transfer, it is present in vitamin B6. And there is tetrahydrofluorate, which is very important for nucleic acid biosynthesis. This is coming for folate or vitamin B9. And thiamine pyrophosphate TPP, which is coming from vitamin B1, which I already told you. Okay, this is help in aldehyde group transfer, and coenzyme may help in acyl group transfer, and flavin adenine dinucleotide help in electron transfer. Okay, and nicotinamide adenine is help in the transfer of hydride ion, not H atom. We have to remember that. So I think you like my teaching. Thank you.